Hi everyone, welcome to a short video on CRC biomarkers. As we all know, our bodies are made of tissues and organ systems, and the basic functional unit of our body is a cell. Inside the cell is the nucleus, which is the command center of the cell and houses the DNA, the genetic material that encodes a blueprint of the entire cell. The tissues and organ systems are composed of tight communities of cells where each cell has specific roles and follow very strict rules to exist as part of the community. A tumor biomarker is anything that can inform us about the tumor. In my talk, I will cover germline testing, tumor testing, and liquid biopsies. Through these tests, we are looking at DNA. And why are we interested in DNA? Normal cells are normal because their DNA is normal. And when normal cellular DNA becomes damaged and changes, it can cause changes in proteins that it encodes. And these cells that have abnormal proteins can behave abnormally and fail to follow the rules in the community that make up the tissue. And this can give rise to a tumor. So first, let us go. And why are these changes important? These changes are important because knowing what is altered in a tumor cell can help us know how to kill it. And clinically, these changes may have important implications and also help us classify the tumor. So let's first look at germline testing. So by germline, I mean the DNA in sperm or eggs. Changes in the germline DNA are heritable and can be found in all cells in the body, including the tumor cells. People with germline mutations may be susceptible to other cancers other than CRC and may need additional screening and surveillance. Since germline mutations are heritable, there may be implications for family, like siblings and children, and they may also need to get tested if an individual is found to have germline mutations. Here is a list of common inherited cancer syndromes and associated genes. Here you can see familial adenomatous polyposis. That condition is caused by mutations in the APC gene and can cause colorectal, brain, stomach, bone, and skin cancers. Another common inherited CRC is because of Lynch syndrome, where you have mutations in these 4 proteins, and commonly it can cause colorectal and endometrial cancers. So, and heritable CRC, that means colorectal cancer because of inherited mutations, are less than about 5% of the total CRC. There are 2 kinds. One is known as hereditary non-polyposis CRC, and that includes Lynch syndrome, which we're all familiar with. And the other one is known as polyposis, which is characterized by, a large number of polyps found in the intestine of affected individuals. I've listed a few genes like FAP, AFAP, or conditions known as FAP and AFAP, which is due to germline mutations in APC. Another condition is known as MAP, or myth-associated polyposis, which is due to mutations in the MUYTH gene. There are other conditions, for example, due to mutations in the POL-E or POL-D1. And another, I've listed these mutations or gene names basically to show that some of the same genes are involved in sporadic CRC, which is what majority of us have. That is the rest of the 95% of CRC, which is not heritable. So next slide. The second kind of inherited CRC is known as Lynch syndrome. And this is due to germline pathogenic variants in the DNA mismatch repair genes, which are MLH1, MSH2, MSH6, and PMS2. So usually Lynch syndrome is due to germline mutations in these 4 genes, and less commonly Lynch syndrome is also caused by mutations in the EpCAM gene. So now that we have seen what germline testing is, let's go find out what tumor testing is. Tumor testing is what, most of us that do not have heritable or inherited colorectal cancer, that is what we all have is known as sporadic colorectal cancer, and we are concerned about tumor testing. So tumor testing looks at changes in the somatic, which means only in the tumor, and it can be, you can find the changes in tumor cells by using very low-tech tests such as immunohistochemistry, that is IHC. And using that, you can look for mismatch repair proteins. You can also look at another common mutation, which is found in about 10% of colorectal cancer, which is a BRAF V600E mutation. So if there are mutations in the mismatch repair proteins, then your tumor is classified as MSI high. And MSI high tumors comprise about 15% of all CRC and about 4% of metastatic colorectal cancer. BRAF V600E mutations are found in about 10% of colorectal cancer. Another mutation that is of interest to most of CRC patients is RAS mutations. There are 3 RAS genes that are important, which are KRAS, NRAS, and HRAS. This is because if your tumor has mutations in the RAS genes, then you are not eligible for treatment using a class of targeted therapy known as EGFR inhibitors. So for anybody who has colorectal cancer, it is very important that they know what stage they are, and if they're stage 4, where their primary tumor is and where the mets are. And then the other important thing to know is whether their tumor is mismatched, the status of mismatched repair proteins. So if you have unmutated mismatched repair proteins, then your tumor is called microsatellite stable, or MSS. And if your tumor has abnormal or mutated mismatched repair proteins, then your tumor is MSI high, or microsatellite unstable high. So this is the basic kind of testing that everyone should have. And there is like different degrees of tumor testing. The more complicated ones, I had my computer freeze up for a second, sorry about that. So the other kind of tumor genomic profiling, which is really extensive and needs more sophisticated equipment, is the tumor genomic profiling. This uses resected tumor tissue, and it looks at various kinds of gene changes. So it can look at gene mutations, deletions, inversions, chromosomal, some kind of chromosomal translocations, amplifications, etc. And many of the newer tests, they will also look at RNA profile, and they give what is known as TMB, which is tumor mutational burden. You can get the value of TMB also by using tumor genomic profiling. There are many tests available. The common ones are Foundation-1, CARES, Tempus, etc. And the most extensive profiling is done by MSKCC. The test is called MSKCC Impact. In these kind of the commonly used tests like Foundation-1, CARES, Tempus, etc., they look at about 300 different changes on over 72 different cancer-relevant genes. So in general, the mutations that are identified, like you'll get a list of mutations, some kind of an alphabet soup. They are in general categorized into 2. One is actionable mutations. That means those mutations that there is targeted therapy against, or for example, with the NRAS and KRAS mutations, if you have those mutations, like I said before, you are not eligible for EGFR inhibitor antibodies. For the BRAF V600E, there is targeted therapy against that particular mutation. For mutations in the MMR genes, then that makes your tumor MSI high, in which case you are eligible for immunotherapy. Some other mutations that are actionable are POL mutations and NTRK fusions, and also HER2 or ERB2 amplifications. So I kind of want to bring your attention here to show that some of the genes that we talked about in the inherited or heritable CRC, those same genes are also seen to be mutated in sporadic CRC. Some of the genes are not, or mutations are not actionable. For example, APC, TP53, etc. These mutations in these genes are important for the change in normal cell to become a colorectal cancer. So right now we don't have any targeted therapies against them. That brings us to the next topic, which is liquid biopsies. So recently I heard this talk from Dr. Marshall, and he called ctDNA tumor trash in the bloodstream. So all cells turn over in our body, and as a result of it, they shed pieces of their cellular DNA into the bloodstream. So tumor cells also do this, but because tumor cells have mutated DNA, because of that property, we can now find ctDNA by virtue of their mutations and then use it as an indicator for the presence of tumors in our body. So ctDNA, liquid biopsies actually look for ctDNA or circulating tumor DNA. And as I said, the assumption is that circulating tumor DNA is abnormal DNA, and therefore we can detect it. So here is a diagram showing the uses of liquid biopsies for treatment in various stages of cancer. In this figure, they're talking about circulating tumor cells, but in colorectal cancer in general, we usually look at circulating tumor DNA, not so much the circulating tumor cells. So using that, you can look at DNA abnormalities, amplification and deletions, translocations, chromosomal abnormalities, and point mutations. And circulating tumor DNA can be used for early intervention. So there are some trials going on to find out if you can look at circulating tumor DNA levels to detect the presence of a tumor. So in say screening. The other use of liquid biopsies is in localized disease to see, to determine the risk of recurrence after treatment. And liquid biopsies are also important in metastatic disease because it can enable treatment selection based on the presence of certain specific biomarkers. The other use of liquid biopsies is to see, once you're on a certain treatment, to find out if there is emergence of resistant clones, and so basically looking at treatment response. So there are two different kinds of liquid biopsies. One is tumor naive and the other one is tumor informed. Tumor naive basically means that you have the same panel for everyone. And example of tumor naive liquid biopsies are Garden 360, Foundation 1 liquid, etc. Here they look at a panel of about 300 common mutations and other changes. The test only needs a blood sample, and the results usually include mutational info. The other class of liquid biopsies is tumor informed. An example is Signatera. In this case, this is a liquid biopsy that is individualized to each person's tumor. So for this, what they do is that you need a tumor or biopsy tissue, and next-gen sequencing of the tumor tissue is done. And then based on the mutations that are present in the tumor, a panel of about 16 different genes are selected for each individual patient, and a liquid biopsy test is designed. So the result of the test, the disadvantage is that it does not contain mutational info. They give you a total value, which is mutations per mil. And it's almost, the result of this test can be almost used like that of CEA. So then in the last slide, I want to go over a comparison between tissue biopsy and liquid biopsy. So with respect to a tumor genomic testing, you obviously need a tumor sample or biopsy. And the disadvantage is that it is a very small portion of the entire tumor. So there is this idea of tumor heterogeneity, which means the whole tumor is composed of a bunch of clones. So when you're sampling only a part of the tumor, and you get those mutations, and you assume that the rest of the tumor is composed of the same mutations, which may or may not be true. And then frequent sampling is not possible with tumor testing because the mets may be in places which are not easily accessible, and you can't go every month to to do a biopsy. And using tumor testing, or you can calculate TMB, and the data that you get from tumor testing is considered historic, not real time. Because if you say, for example, a surgically resected tissue, then that tissue is no longer in your body. So it's giving you historic information. You compare liquid biopsy, it does not need a tumor biopsy, except in the case of like a tumor-informed test such as Signatera, where you need, for the very first test, you need a sample of the tumor. A liquid biopsy is supposed to be more representative of the whole tumor, because it is thought that different parts of the tumor can shed ctDNA, and so therefore, when you look at blood to find ctDNA, then you assume that you're getting a representation of the entire tumor. Frequent testing is possible because all you need is a blood draw. For now, a liquid biopsy cannot be used to calculate TMB, but maybe in the next few years, maybe it may be possible to calculate TMB from liquid biopsies. Serial liquid biopsy testing can give you real-time info on response to treatment. So for example, if you did a liquid biopsy before you started treatment, and then you got on like a targeted therapy like EGFR inhibitors, or if you have a BRAF mutated tumor, then you are on targeted therapy against the BRAF mutation, then over time, if the tumor starts overgrowing, you can do another liquid biopsy and then see if, what are the mutations? Are there new mutations? And is that the mechanism of how the tumor has become resistant to this particular treatment? So this is a direct comparison between, how the differences or advantages, disadvantages of tumor testing versus liquid biopsy. So with that, I'm going to stop sharing my screen. So the idea is that this video will be deposited in a common place, and that everyone who's part of the EPL can, watch it at their own convenience. And during our first session, I hope to answer any questions you have. And like I said before, in my introductory video, there will be a short quiz. So I hope you guys find this video informative. So thank you very much. Bye.